Hi everyone, this is Jason Key at SB Grid at Harvard Medical School in Boston. Today I'd like to welcome Tom Grant from the Hauptman-Woodward Medical Research Institute in Buffalo. Uh, Thanks for joining us, Tom. And Tom today is going to tell us about small angle scattering. Thanks, Tom. Take it away.
All right, great. Thank you, Jason. I appreciate you all taking some time to listen to this. I'm going to talk today about small angle X-ray scattering, well, actually small angle scattering in general. And what will happen is I'll break this up into two parts. So today's part will be an introduction on the theory and practical applications in terms of data quality analysis. And then next week we will talk more about the advanced methods of small angle scattering. So my email there is if you have any questions, you can feel free to send me an email and I'll see if I can do my best to answer them. So first I want to talk about just some quick literature and software for you to be able to go to if you have questions that'll be able to give you a lot of information that are both broad and specific depending on where you go. So the first one is the review that was posted previously online at the SB Grid website, and it's the Putnam review from 2007. It's an excellent review. It's very broad. It's about some 90-page review or something like that, so it's quite extensive. The Jacques and Truella review from 2010 is very good for practical considerations when dealing with SAXS data. Svergan et al. Came out with a book this past year that's a very extensive analysis and of both practical applications and software applications, as well as the theory of small angle scattering, particularly from solutions of biomolecules. Additionally, I just want to list a software website where you can get a lot of the different software that's available for small angle scattering analyses for both biological and non-biological applications. It's just kind of a general website for small angle scattering. However, for biological applications, the most common package appears to be ATSAS, which you can find on that website listed there, and that's usually what publications will most commonly have used for their analyses. However, there's a lot of other excellent software packages that exist too. All right, so I first want to talk about what is small angle scattering. So this is a molecular transform. If you're familiar with crystallography or really any kind of diffraction, you'll probably be familiar with what a molecular transform is. So molecular transform is the Fourier transform of the electron density of a particle. Okay, so this is sort of the equivalent of what you would see if you took the X-ray scattering or the X-ray diffraction pattern from a single molecule. If you had the technology to be able to do that, this is sort of what you would see. This is just one angle. So if you actually think about crystallography, which many of you are going to be crystallographers, I'm sure, that are watching this, but we use Bragg sampling. So you have an ordered array of the molecules arranged in a certain symmetry packing conformation, and what happens is that causes reflections to occur, and so you get certain angles where the equations are satisfied such that you'll actually see coherent X-ray diffraction in certain spots, and so that's overlaid on the bottom left here. And so what happens is when you're actually doing X-ray crystallography, what you're doing is sampling that molecular transform pattern. So you end up with a series of reflections with different intensities, where the intensities are determined by the molecular transform, which is itself determined by the arrangement of the atoms within the molecule, whereas the positions of the reflections are determined by the packing of the crystal. Well, from small angle scattering perspective, we actually still start with the molecular transform. We're getting the same information, we're just sampling it differently. So in small angle scattering, instead of looking at the entire molecular transform out to, say, a resolution of about 2 angstroms, what we're actually doing is just zooming in on the small angle region, hence the name. And generally it's about maybe to 20 angstrom resolution. So generally what you do in small angle scattering is you don't see a pattern that looks like this, though. You actually see a isotropic pattern. So this is an anisotropic pattern from an actual molecular transform, a simulated molecular transform. But what happens is because we're doing solution scattering, you actually have spherical averaging. The molecules are tumbling in solution and they're tumbling over the time of the experiment. And so you end up with isotropic scattering on your detector. And so you lose all that information that's available in the 2- and 3-dimensional patterns that you don't get in an isotropic pattern. So what happens in a basic SAXS experiment is that you have incident X-rays that impinge upon a sample with the solution and the molecules tumbling in solution. And so there's a spherically averaged intensity that's recorded, and so it's an isotropic pattern. And what you do is you have a radial integration of that pattern, and you end up with a one-dimensional profile. And so this is reciprocal space. So what happens is that larger particles scatter at smaller angles, and smaller particles scatter at larger angles. And so the analysis of this 1D profile is what gives you your information about the size and shape of the particles that are in your solution. So what's important in SAXS is contrast. So SAXS is inherently a contrast method. What that means is that it depends on the square of the difference in the electron density between the molecule of interest that you're studying and the solvent that's surrounding it. And so this is just a little graphic to show you what kind of a comparison that is. And so if you think of like a protein, the electron density of a protein is somewhere around, say, 0.44 electrons per angstrom cubed. And if your solvent was, say, water, your background is about 0.33 electrons per angstrom cubed. So when you actually work that out, your scattering signal is very small. It's only about 10% above the solvent scattering itself. And so you're looking for a very small signal when you're looking at SAXS because of this contrast issue. So in order to assess that background solvent and pull it out, that bulk solvent scattering, we actually measure two signals when we do a SAXS experiment. You measure the total protein plus buffer, which is shown in blue, and you also measure the buffer signal by itself without protein. And that includes any kind of other background scattering, such as the capillary or the flow cell, whatever the experimental setup that is you're using, and you subtract it from that total protein plus buffer scattering, and that gives you your final scattering pattern of the protein. So once you have that, we can kind of break that up into a few different regions, okay? So we have a plot here of the log of the intensity on the left, on the y-axis, and we have Q on the x-axis. So Q is essentially the angle, okay? So it's a transformation of the angle where the wavelength is involved. But you want to make sure that when you're looking at Q, that you look at whatever publication is that you're referring to, or if you're writing a publication, that you make sure how Q is defined is written in that paper. And so that'll be very important because different applications will use, say, a term called S, which is almost interchangeable with Q, but it's actually a little bit different by a factor of 2 pi. So you just want to make sure that you're clear when you're reading publications or when you're publishing yourself, that you're clear on that. So to break up a SAXS curve, generally we have 3 different regions, qualitatively speaking. And so the low Q region deals with information about the size of the particle, whereas the middle region sort of deals with differences in the shapes of particles. And the higher Q region deals with internal structure, which means you can get information about flexibility and solvent exposed regions and stuff like that. All right, so what can SAXS provide? So first of all, the SAXS scattering pattern can provide a radius of gyration of your molecule of interest. It can give you a maximum particle dimension. I should say what a radius of gyration is, just in case anybody's unfamiliar. So a radius of gyration is the square root of the sum of the squares of all of the distances of all the atoms to the center of mass of the particle. So it does change depending on the shape of the object. Additionally, it can give you the maximum particle dimension. It'll also give you information about the molecular weight, and from that you can actually get the oligomeric state. And not only can you get the state as like a dimer versus a monomer, you can actually get the organization of the oligomer in solution, how they're packed relative to each other in that oligomer. Additionally, you can get information about the native flexibility or unfoldedness of a particle. You're able to actually visualize regions that are disordered in electron density reconstructions from X-ray crystallography. So in X-ray crystallography, atoms have to be ordered very, very well, pretty much to within an angstrom between the different unit cells. And if they're not, it enhances your B factor, it increases your B factor, which causes your signal to decay to the point of not being seen in your electron density. Whereas because SAXS is a low resolution technique, you can actually see some of these regions that are disordered in either your envelopes or your modeling. And finally, what we'll talk about today is some modeling of low-resolution molecular envelopes. So that's probably one of the most common things people think about when they think of SAXS, is low-resolution molecular envelopes. So there's a basic equation when it comes to what you're actually recording on your detector for the scattering intensity. So the scattering intensity that you record is equal to the form factor of the particle, F, times the structure factor of the solution, S. So the form factor describes the intraparticle interactions, sort of the atomic distances within the molecular envelope, and so that can give you information about the size and shape of your particle, whereas the structure factor describes interparticle interactions. So if you've got repulsion or attraction in your solution state, then that's actually going to contribute to your intensity. So when we're looking for information about size and shape, we want the form factor. And so we use the intensity to get at that. So what we need is the structure factor to equal 1, i.e. that there is no interparticle interactions present in the solution. So to give you an idea of how the structure factor affects the scattering pattern, on the left here we have the form factor. Okay, so on the top we have an example of a structure factor where a solution is attractive, and on the bottom we have a solution where the particles are repelling each other. And so you notice that the structure factor in red for the attractive molecules. Hey Jason, I'm just going to pause for a second. Am I able to, can you see my mouse?
Oh, yes, I can see your mouse.
Oh, okay. So on the top we have the attractive structure factor, and in red you notice that at low Q values the structure factor is above 1. It's increasing the apparent scattering. And so what happens is when you multiply those, what you actually would record would be an increased low Q value compared with the form factor, and it's just the opposite for repulsion, where you actually end up with a decreased low Q values for compared to the form factor. And so what that causes is, in attraction, it would mean that your Rg is larger than it should be. The calculated Dmaxes will likely be larger, the volumes will likely be larger, the corresponding molecular weights will be larger, and all your numbers are just going to be off. Additionally, for repulsion, it will be just the opposite. You'll get smaller Rgs than you would expect. You would get smaller volumes, smaller molecular weights, and smaller size parameters. So that's why we want to make sure that our structure factor is, in fact, 1 in all of our cases. In other words, we want to make sure that there are no interparticle interactions occurring within our solution. And this is probably one of the most important things when it comes to preparing your samples for SAXS data analysis. So one of the ways that we can look at whether or not there is a structure factor or attraction or repulsion within our scattering pattern is something called the Gagne method. So this was developed by Andre Gagne back in 1939. He's kind of like the founder of SAXS. And he was able to come up with an equation that approximates the low Q data, as Q approaches 0, that you can approximate the intensity by this simple equation. And so what happens is if you take the natural log of both sides, you end up with something that actually looks like a line. So if you plot the natural log of I versus Q squared, you end up with a linear regression. So if you actually plot that in what's called a Gagne plot, what you should see is your data should be linear. And so this can actually tell you something about the structure factor of your solution. So if your data is not linear, say it's got a positive curvature here, what that means is that you've got some kind of attraction or aggregation in your data, and you don't want to be processing this data. You want to go back to your drawing board, your solution state, either throw some salts in there or figure out what it is that's causing your solution to be attractive or aggregated, and fix it so that you can go back, redo your experiment, and make sure that you have good quality monodispersed data. Similarly, if it were repulsive, you would actually see, instead of the smile, you would see sort of a frown. Okay, so you would see negative curvature there. And these green points here are the residuals, which can help you to identify that nonlinearity. So when it comes to the Gagne region, I've sort of just so far said that you use low Q data. Well, there are some quantitative values that you can apply to that. So when you collect your SAXS data, it's very important that the lowest data point that you collect meets this condition. Okay, it's got to satisfy this condition, and that condition is that the Q value of that minimum data point is less than pi divided by the maximum dimension of your particle. And the reason for that is based on information content. If you're not collecting angles, scattering angles that are smaller than the largest dimension of the particle, then you're actually missing information that your particle is scattering, which in terms of SAXS data is very important. So in crystallography, a lot of times you can get away with not collecting your 001 reflection. Nobody ever does that. But in SAXS, it's very, very important that you make sure to collect data less than pi over d max. Gagne's equation is only valid in low Q region. So they have sort of this kind of rule of thumb that seems to be in the literature that Qmax, where Qmax is the maximum Q value of your Gagne region, is less than 1.3 divided by Rg. This is not a hard and fast rule. This is just kind of a rule of thumb. Depending on the shape of your particle, if you have a very elongated particle, you're going to have a smaller region. So it's generally around 0.8 divided by Rg. And so who you talk to kind of varies on whether or not this is 1 or 1.3, but this is just kind of a rule of thumb. Globular particles will tend to agree with the Gagne region to a much larger extent or to a much higher Q range than will very anisymmetric particles. So one of the things that's very important is that since the region here is dependent upon the size of your particle, you need to make sure that when you have very large particles that you pull your detector back far enough. And the reason for that is that if your Rg is very large, then you might only get 5, 10 data points in your Gagne region. And if you have too few data points, then that makes your Rg estimate very poor. And you want to make sure that you get as good of data as you can, obviously. So you want to pull that detector back to make sure you're collecting your data to the lowest possible Q value that you would need for the size of your particle. So once you've assessed that structure factor using your Gagne plot, and you make sure that you've got no interparticle interactions, the next thing to do is to take that intensity data, which we now know as your form factor, which contains all the information about the size and shape of your particle, and get that shape and size information out of it. So one of the ways to do this is to use a Fourier transform. So if you take the Fourier transform of your intensity profile, you end up with the real space representation, which is called the pair distance distribution function, or P of R. And so immediately from the P of R, you can get some information qualitatively about the shape of your particle. In the case of some kind of shape like this, where you have kind of a Gaussian bell curve looking P of R curve, what that means is that you have a pretty globular particle. It's relatively spherical, not too many features. Whereas if you have something that's left skewed, that means you have an elongated particle. If you have something that's very right skewed, for example, if you had a virus capsid, you would end up with a very right skewed curve that would extend up and then drop down straight to 0 there. What we use actually instead of a direct Fourier transform is something called an indirect Fourier transform, and this is the basic form of the equation that satisfies. So there's some software that will perform these for you, and so this is how you can get your P of R curve. So there's a few things that you can get from your P of R curve. And one of the first things that people get is the Rg. So I mentioned the Rg a second ago that you can get from the Gagne plot, but you can also get your Rg from the pair distance distribution function. There's a few advantages to using this method too. First of all, it uses the entire curve. Because you're using the Fourier transform of your entire intensity profile, you're getting information that is less sensitive to interparticle effects. So if you remember, the Gagne region uses only low Q data, but also the structure factor for interparticle effects most affects low Q data. And so by using the pair distance distribution function, you can get a really good estimate of Rg that a lot of times is more accurate than the Gagne estimate. This is especially useful for large particles with a very narrow Gagne region that only has a few data points, or with noisy data. It's always a good check to make sure that the two Rg's match, that from the Gagne region and from the P of R distribution. Additionally, you can also get the maximum particle dimension from the pair distribution function. So the P of R should gradually fall to 0 at Dmax, and how gradually it falls to 0 is going to depend upon the shape of your particle. But you shouldn't get like abrupt termination. So if you look at this example on the right here, for this protein, the Dmax is somewhere around 100. But if you notice that if you chose, say, 70, when you're estimating this in your software, it appears to have this forced abrupt descent to 0. And so that suggests that it really kind of wants to go out to a higher R. Whereas if you give it a large Dmax, it kind of falls off at around 100 and then just kind of oscillates around 0. And so what that suggests is you want a smaller Dmax. And if you give it 100, you notice you get this nice gradual decay to 0. So this is kind of a nice practice of kind of how I do it when it comes to looking at what my Dmax is. I'll just give it a large Dmax, and I'll find out where does it naturally fall to 0. And then I'll recalculate it and iterate through some values to find out what the best Dmax is. Now, one thing to keep in mind is that Dmax is somewhat of a poorly determined value. So it can be, even for good data, it can have errors of 10 to 20%. For example, here, I could probably choose anywhere between 85 and 105 and get somewhat of a similar P of R curve. Okay, so you can see that there's some large errors simply because it's not a very well determined value. So these are some of the ways that the sample quality, those interparticle interactions, can affect your data analysis. And this is from the Jacques and Truella review that I mentioned earlier. This is a really excellent review when it comes to practical applications and practical analysis of your data. So I believe this is lysozyme, a lysozyme scattering profile. And so on the left here we have what good data is. Okay, you notice we've got a nice linear Gagne region. Our RG is 14.7. When we look at our P of R curve, it decays nicely to 0 without any forced decays. And so we get an RG of 14.3 that agrees pretty well with our Gagne RG. And additionally, when we look at a large Dmax, we get a pretty well-defined actual Dmax of about, say, 45 angstroms there. Okay. In the case of repulsion, some kind of interparticle interference here, we've got both subtle and severe. You notice that for the severe case, you see this this sort of negative curvature in the Gagne region that I mentioned before. You also see a substantial decrease in the apparent RG from your Gagne plot, from the slope of that line. And so you also get a decrease in your Dmax. So instead of it being about 45, maybe for what it is for the good data, you see that it's about 30 or 5 or so, right? And so you get this decrease in Dmax. Additionally, if you were to test out high values of R, you actually end up with negative P of R values, which you shouldn't have negative P of R values for any kind of protein sample that you have. In the case of attraction or aggregation, you'll actually see increases in your values. So you notice the RG is substantially higher. Additionally, your Dmax is very difficult to determine because it just keeps extending. No matter how large you give it, it essentially just keeps extending. So it's a much larger Dmax. And so you want to make sure that, again, your RGs from both methods match up pretty well. You notice that in this case, you get an RG of 21.6 versus the actual RG of 14.7 about. So this all goes to data quality. The most important thing in SAXS, which is obviously always important, is data quality, making sure you characterize your sample to ensure there's no aggregation. So one thing I want to point out in this previous slide is that even for a slightly aggregated Gagne plot, it still looks pretty linear, right? And so the Gagne plot is not the end-all be-all of what defines whether or not your data is good, okay? It's just really good at assessing when you have severe effects, okay? Like in this case here, and in the case I showed you earlier when there was some severe aggregation, those are pretty apparent. But that's not always the case, okay? And so you want to make sure that you characterize your sample with other methods, either through static or dynamic light scattering or gel filtration or some kind of mechanism to make sure that you have no aggregation or polydispersity in your sample or any kind of interparticle interactions. Generally, you want to use as high of a concentration as you can while making sure that you don't have those interparticle interactions. So higher concentrations yield higher signal to noise. And with SAXS, that's obviously very important. So typical concentrations, practically speaking, are generally around 1 to 10 mg per mil. Smaller particles, because they scatter less, will require higher concentrations than larger particles. Additionally, as I mentioned before, this is a contrast method, and so RNA and DNA have larger scattering cross sections than does protein, and so you don't need quite as high of a concentration because they'll scatter substantially more. Whenever you have higher concentrations, you want to make sure that you check multiple concentrations to ensure that there's no concentration dependence occurring. Additionally, while high signal to noise is important, it's not quite as important as good sample quality. You can have really good signal to noise, but if you've got poor sample quality, then that all goes to nothing. Whereas you sometimes can get information about poor signal to noise about your sample from data that has poor signal to noise, as long as it's a good sample quality. And additional to all this is that you need to make sure that you have an accurate buffer subtraction. And buffers can actually differ quite substantially in their scattering profile. So you want to make sure that your scattering, or that your solution, your buffer solution in your protein sample is identical to the buffer solution in your buffer blank that you're going to collect and subtract from your total protein scattering. Usually that can either be done pretty well either through dialysis or some kind of column flow through buffer. So there's multiple methods to estimate the molecular weight. I mentioned this earlier that from the I0 that you can get I0 through the extrapolation. Well, what you can use with that I0 for is to estimate the number of electrons in your particle, which can tell you your molecular weight. Usually the way this is done practically is through some kind of standard, either a protein standard such as BSA or xyloisomerase or lysozyme, something that is known to be monodisperse, easy to work with, cheap, and is monodisperse at higher concentrations. And so the main problem here, the main source of error when it comes to determining molecular weight this way, is the concentration. In order to use I0, you have to know the concentration of both your protein and your protein standard. And so because of this, sometimes people actually will use water. Water has a known scattering based on Thomson scattering and the scattering of the electron. And so you can actually use water scattering to calibrate your curve. It's a little bit more involved, involves transmission coefficients, but if your beamline will set that up for you, it shouldn't be too bad. But that removes the source of error of concentration for your protein standard. And so you want to make sure that you know your concentrations as accurately as possible. You can also get your molecular weight by knowing what the particle volume is. So the SAXS will tell you information about how much volume, how much space the particle occupies in solution. So if you use the fact that density equals mass divided by volume, then you can see that if you assume, say for proteins, a density of 1.37 grams per centimeter cubed, then you can use that to estimate what the molecular weight is if you assume a uniform protein density. Some problems with this arise, however, because some particles are disordered, and so they don't have a defined volume that they occupy in space. And so if you have a disordered particle, it might actually look like it's occupying more volume than it really is, simply because it's an average. So actually last year, a new method came up by Rob Rambo and John Tainer. They found that it was actually accurate even for disordered proteins. And so that's a great tool. Actually, I find that it's even at least as accurate as the particle volume method for regular globular proteins, not just unfolded proteins. There's another method called SAXS-MAO that uses calibration with the protein data bank. And so they have a little Java applet online that you can run your data with, and it'll give you a pretty decent estimate of your molecular weight as well. So there's multiple avenues to get your molecular weight. However, all of these methods are generally only accurate to about 10% or so. You know, that's about as good as it gets. And so what this means is that if you're looking for differences in molecular weight, say for example you have a protein-protein complex that you're looking at, where one component of your complex is maybe 100 kilodaltons. If the other component of your complex is only 10 or 20 kilodaltons, it's going to be very difficult to determine whether or not you've actually formed your protein complex because the molecular weights are so similar. So you really want to make sure that you have enough signal noise in that case to make the best possible case to compare, some existing models. So I mentioned about a second ago about the difference between folded and unfolded particles. Now when it comes to folded and unfolded particles in solution, they have different scattering profiles. So what we're plotting here is the intensity times q squared versus q. Okay? So again, this is just an example of how S and Q are somewhat interchangeable. But this is called a Kratky plot. And so a Kratky plot will be globular, or will be, for a globular protein, will show this blue curve. Okay, and so what will happen is you'll get kind of a Gaussian bell curve type thing where you have a peak and it decays back to zero. Okay, and so that shows that your particle is folded. Whereas if you have something that is unfolded, you won't have a peak. And so what will happen is it will just plateau and keep increasing like that. Okay, so that suggests that you have an unfolded particle. If you've got a partially folded particle, you'll actually kind of have a combination of the two. Okay, so you might have a peak, and then it might not dip all the way back to zero, but it might go a little bit down and then continue back up. Okay, so that might say that you have a partially folded molecule. If you have a folded molecule, but it's flexible with, it's got multiple domains with flexible linkers, you might see something more akin to this red curve here, where you've kind of got some shoulders on your curve, but it still decays down to zero, suggesting that it's folded. So that's how you can get qualitatively some information about flexibility within your system. So now we're going to go into a quick overview of envelope reconstruction. This is kind of going to be the only type of modeling that I'll talk about today. So envelope reconstruction is probably one of the most common things that people think about when they think about SAXS. Usually if you publish something, they always want to see an ab initio envelope. And so what generally it means is that you kind of, in the case of bead modeling, which this example here is bead modeling, there's a lot of different software packages that will do ab initio modeling. The most common seems to be DAMIF, which is from the ATSAS suite. And so generally the way it works is they fill a volume of space with a grid of beads. Okay, and so we assume that particle density, the protein or whatever, is uniform within that space and within its envelope, and all the rest are solvent. Okay, and so what it does is it starts with a random configuration of beads that are solvent and beads that are particle, and it tests its scattering pattern, it calculates it and compares it against your experimental data, and then it changes them. And beads that help your scattering pattern, if beads of being particle helps your scattering pattern, then it will keep those beads, and ones that make your scattering pattern worse or less like your experimental data will reject those. And so unfortunately what happens is that if you imagine for a second that you've got, say, 2,000 beads, to test all the different permutations would be 2 to the 2,000. And so that's way too many computations to actually do, even in the age of the universe with every computer we have. So we use Monte Carlo-like approaches to sort of sample different to sample the configurational space. However, the problem is that they can easily fall into local minima. And so what happens is we use simulated annealing to help find a global minimum that uses random seed generation. So the problem with this is that you can still fall into its local minima, and so it's very, very important that you avoid over-interpreting your envelopes. Envelopes are very nice pictures. They're great to generate hypotheses and to possibly validate some largely discrepant models. But you want to make sure you don't over-interpret envelopes. It's very easy to do. So the way it is able to make protein-looking envelopes is that it doesn't just randomly search configuration space. It actually uses some penalties. So if you think about extracrystallography or NMR or anything like that, you're not just using your raw data most of the time. Usually you're incorporating information about, say, bond lengths or Ramachandran plots, torsion angles, other chemical information to help introduce that information, those restraints, into your modeling process so that that information can be used to give you a better model. And so DAMIF does something similar, and any bead modeling program for the most part will do something similar. but we don't have that kind of high resolution data such as chemical bond lengths. But we do know certain things about protein envelopes. Generally, they'll be connected. You have, for a single chain, you have one long primary sequence that's going to be covalently connected to itself. So you're not going to have some, chunk of protein over here and some chunk of protein over here. It's going to be one molecule. And so different penalties like that are incorporated into the modeling software, and it gets combined into this score function. So one is obviously the fit to your data, and another are the penalties that are associated with it. And so what happens is you end up with these somewhat protein-looking envelopes. Now, because of the simulated annealing protocol that I mentioned, you start with a random seed each time you run this. And so you'll actually run this multiple times. If you run the same program twice with the same exact data, you'll actually get two slightly different answers. So what we do with that is we actually use averaging. So what you'll do is you'll generally create 5, 10, 20 different ab initio envelopes, and you'll average them together. And so you notice that while each model sort of has the same general shape, there's some features that are different. There's some there's some beads in certain regions that are not present in other regions and other molecules and other envelopes. And so what you do is you average them. You find out what features are common among all envelopes that are more likely to be a result of the data rather than features that might be only a result of the random seed configuration. And so you use this averaging to build up a consensus envelope that is then more likely to actually represent what is in solution, what your particle actually looks like. So I'm just going to show a few examples now of some different work that we've done in our lab. So this is actually from a few years ago. This is part of my graduate thesis. And so what this is a two-domain protein. And this is actually one domain of that two-domain protein. And we were unable to crystallize it, the whole thing. So we used SAXS. It was actually disordered, whereas the C-terminal domain was well ordered. This is the N-terminal domain that was disordered. And so we actually cut off a fragment of it and looked at it with SAXS, and this is what we found. So we didn't have the structure at this time, but I was able to subsequently crystallize it and determine the structure of it, and it turned out to overlap very, very well with what was in our SAXS envelope. So to show you a space filling configuration, you can see that when it comes to SAXS envelopes, you got to keep in mind that this is low resolution. So there's certain regions you're going to notice that are not quite filled. There might be a couple of atoms popping out the side or something like that. But generally, you can see that the envelope fits quite well. Now, there's certain things that maybe are a little bit too large, like this big space over here. It's possible that should actually be filled by this linker. Okay, and so it turns out that that's likely to be the case. This linker here was actually forced by crystal packing to be in that position. So SAXS can actually tell us little bits of information like that. That's kind of the borderline. You don't want to get too much into too high of resolution information like that. But the point being that you want to make sure that your structure matches your data, matches your SAXS envelope in solution. This is another example of a structural genomics consortium target. And that was our envelope that we got before we got the structure, and then we got the structure, and there was some extra beads over on the side here. And we found out that there was in fact 12 residues that were not resolved in the electron density of the X-ray structure. And so that actually helped us to orientate it. And so one of the things you'll notice is that if you have more anisymmetric envelopes or anisymmetric particles, that's very useful in terms of SAXS. If what you're looking at is essentially a glob, like a sphere, then it's going to be difficult to orientate your particle in solution in your envelope. Whereas if you have some defining features, then that can actually be very helpful. So something that's anisotropic like that can actually be very, very useful in terms of determining the orientation relative to the envelope. This is another example that we had from the structural genomics group. We got the structure after the fact, and additionally, there was a large region of about 50 residues that were not resolved in the electron density, but we actually see them in our SAXS envelope. And so there are certain regions that you can, sorry about that, there are certain regions that you can see in the, hold on one second, we're going to have to pause this. Hey, I'm doing a webinar right now. Can I call you back? So as you can see, there are certain regions within the envelope that are visible that you can't find in your electron density. So SAXS here can actually give us additional information that is not available in X-ray crystallography. So just one more quick example. This was a case where we had a structure of a protein that was unknown, and it still hasn't been solved. But there is actually a homolog that's available in the PDB, and it turns out that the dimer of that homolog fits very, very well. And this is a dimer defined by the symmetry mates from the crystal packing. And so that pieces like this, you can actually find out what the oligomeric organization is. This was additional information that the structural genomics people working on this project didn't know originally. So we actually have the organization and solution. It can give you some information about what solution interactions are actually occurring when you can use information from homolog structures as well. All right, so that's about it. I'm going to start wrapping it up now. But what I kind of want to focus on is that it's really important that when you're doing SAXS, you have a question that is defined. Okay, some people have come to me in the past and said, I want to do SAXS, and I'll say, why? And they'll say, because I want an envelope. And I'll say, well, why? And they're like, well, because I can't get my structure from crystallography and from NMR. I'm like, okay, so what is SAXS going to do for you? Okay, so having an envelope obviously is useful, but an envelope in and of itself is really not the purpose of SAXS. Okay, it's useful information from SAXS, but you want to have a question that SAXS can actually provide an answer to. So you want to make sure that the question drives how your experimental setup is determined. So if you've got a case where you've got a very large particle, then you want to make sure you pull your detector back, right? So you get a large sample to detector distance in order to get those low resolution Q values. You also want to make sure that if you've got very small differences that you're looking for in the high resolution region, that you pull your detector up as far as you can, or maybe offset it, so you can get those high Q values to get your high resolution differences. Because maybe the only differences, say you've got two models, maybe the differences in those two models only are discernible at high resolutions. You want to keep in mind that when you're determining your sample to detector distance ahead of time, maybe just make some quick estimates. You've got your primary sequence. Maybe you can say, okay, I know that this is only 10 kilodaltons. You don't need to pull your detector back that far. But if you've got the ribosome or a virus particle, then obviously you need to pull it back as far as possible. In terms of complexes, it's very important to know what the molecular weight difference is between the complex and the proteins themselves. And the reason for that is because, as I mentioned before in that example, if you've got a very small difference between the different complexes, say 100 kilodaltons versus 110 kilodaltons, that's going to be very difficult to look at that and define anything with certainty because the difference is so small. So you want to make sure in those cases that you get both your low Q data that you need and also the highest resolution possible to be able to distinguish between different models. This is a solution scattering experiment, and so it's very important that you know what is in your solution. If you've got different buffer conditions in there, such as higher salts or different additives, you want to make sure that those are present in your buffer blank. Additionally, you want to know how many different concentrations you need to do. If you should do serial dilution. If your protein doesn't behave well at high concentrations, then maybe you need to think about maybe some more exposure time rather than a higher concentration. One important thing to think about is in terms of signal to noise is that exposure doesn't propagate the same in terms of signal to noise that concentration does. So if you do twice the exposure, you won't get twice the signal to noise. And so some possible options for that are that maybe you can make lots of small volumes and shoot each of the small volumes. The problem you have to think about is radiation damage. So obviously, the more you expose it, the more likely you are to get damage. So you want to keep that in mind. So just for a final summary slide, you want to make sure that in all cases that require information about size and shape, that your SAXS sample is monodisperse. You want to make sure that you have the best quality data you can before you try to draw your conclusions. As I showed you, it is very difficult, or rather very easy, to draw conclusions that are based on improper modeling. Okay, it's very easy to overinterpret envelopes or different models. The term resolution in SAXS is somewhat ambiguous. You might think of it like in crystallography, it's 1 over d or 2 pi over q in our case. And whereas you took out your high Q data to maybe 20 angstroms, that doesn't necessarily mean that your model is a 20 angstrom resolution. It's somewhat of an ambiguous term because of the spherical averaging. You're losing that information. Really what resolution is in terms of SAXS is the ability to discriminate between models. And so while useful, you don't want to read too much into envelopes. And I'm going to show you a quick example of that in a second. SAXS is a solution technique, so what's in solution is very important. If you've got temperature variation or pH sensitivity, you can actually use this as a very powerful technique to say, how does the conformation of my protein change, how does the complex formation change as a function of pH, or say I throw in ATP and I see that my protein folds or it changes conformation. This is one of the most powerful things that you can do with SAXS, is to screen different solution conditions. You want to make sure that in all cases you back up any conclusions that you draw with some kind of other experimental evidence before publishing SAXS data. You're not going to be able to publish a SAXS paper that has just SAXS in it for the most part. You're going to want some kind of other information in there that corroborates your SAXS data. So this is an excellent example of what SAXS can do for you that I got from my PhD advisor, Eddie Snell. And so this is just a typical SAXS pattern, and it's possible to fit multiple envelopes to the data. Okay, so what happens is that because of that, you saw a little bit ago that just running the same exact data in the modeling algorithm will give you slightly different ab initio envelopes. Well, in this case, we got this envelope. It's pretty globular, nothing terribly interesting about it. But this is actually from this data on the right. And what's very interesting is that this data is actually Dow Jones closing values from August 2007 to March of 2009 as the 2008 crash was going on. And so what's important to note here is that you will always get an envelope despite the data. Whereas in, say, crystallography, if you have a wrong model, then you're going to have R factors that don't go down, right? They're always going to be high. You might not even be able to phase and come up with any kind of solution with crystallography. Whereas with SAXS, you always, no matter how bad your data is, no matter how noisy it is, no matter how aggregated it is, you will always get some kind of envelope. And so it's important to keep this in mind when you're doing your SAXS experiments. All right, so with that, I just want to invite you out to the part 2, which I'll talk about some more advanced methods in terms of what you can do with your SAXS data beyond simple ab initio modeling and size and shape parameters. You can do rigid body modeling, which is how you get information about complexes, ensemble analysis and flexible systems for, say, disordered proteins. You can get information from mixtures, and you can also do some more advanced protein-protein docking. Additionally, you can do structure refinement. You can include NMR information to do hybrid modeling. And finally, we'll talk about some neutron scattering and contrast matching, which is a very powerful technique that uses neutrons instead of X-rays. Most of the stuff I've talked today about has been focused on X-rays, but neutron scattering is also very powerful, and the theory that I talked about is still just as applicable for neutron scattering as well. So with that, I invite you out to next week, and I thank you for your time.
Great, thank you very much, Tom. And we will be adding part 2 to this series shortly, so check that out. That should be on in about a week. So if you're coming to this a little later, it may already be available. So take a look. Thank you very much.